My name is Anna. I'm an FASD Prevention Program Coordinator with the Saskatchewan Prevention Institute. And as I said, I live in Saskatoon on the homeland of the Cree, the Dakota, and Métis nations. I'd like to take this time to honor this and all Indigenous nations from Turtle Island who have been the caretakers and one with this land for millennia. We are very happy to partner again with CanFASD to bring to you this webinar on brain and behavior changes with low PE, with research findings that are fresh out of the open, have been published really recently. So I have just a few housekeeping items before we get going. We will be recording this presentation. And later it will be available on CanFASD YouTube channel and the SPI website. Participants, because this is a webinar, participants won't have like video or sound, but you can ask your questions at any point of the webinar using the Q&A box. And during the presentation, the presenters may answer during or after. By the end, I can pose your questions from there. So please feel free to drop your questions there. So now I'd like to quickly introduce both presenters, Dr. Lebel and Dr. Long. Dr. Lebel is an associate professor of radiology at the University of Calgary and the Canada Research Chair in Pediatric Imaging. She leads the Child, Brain and Mental Health Program at the Alberta Children's Hospital Research Institute and is a member of the Hotchkiss Brain Institute. She received her PhD in biomedical engineering from the University of Alberta and completed postdoctoral training in neurology and pediatrics at the University of California in Los Angeles. Her research uses MRI to study how brain structure and function changes function change with age in typical children and those with neurodevelopmental disorder, including FASD and learning disabilities. She also examines how brain structure and function are related to cognitive, behavioral, and environmental factors, including the prenatal environment. Dr. Long is a research associate in the lab of Dr. Lebel. He obtained his PhD in Germany and then completed a postdoctoral fellowship with Dr. Lebel. His research focuses on the use of advanced MRI to study typical brain development and brain differences in children with FASD. I have to say that as soon as I learned about your findings through the CanFASD newsletter, I looked up your contacts to contact you because evidence of harm from low exposure to alcohol prenatally, it's not that easy to come across, and conflicting messages about just a little bit of alcohol during pregnancy are not rare. So yeah, I'm very happy that you could join us, and yeah, I'm happy to pass to both of you.
Thank you very much for the introduction, and thank you for having us. Yeah, we are also very glad that the CanFASD really interested in this study and invite us for this webinar. And also I'm happy to share like a lot of details and studies, everything from this study and also from the whole lab. So yeah. Welcome to this webinar. So you can see the title is talking about the brain and behavior change in low amount of prenatal alcohol exposure. And we are from Calgary. We're from the University of Calgary. So anybody from Calgary could say hi here. So. And for the disclosure, we don't have any conflict of interest. The paper is and both of us, like two of us, there's no conflict of interest of anything. And also at the very beginning, I'd like to take this opportunity to acknowledge the traditional territories of the people of the Treaty 7 region in Southern Alberta. And also the city of Calgary is home to Métis Nation of Alberta Region 3. Okay, so this is the outline of my presentation or webinar today. So first I'm gonna go through some background, like what we are doing and what the study used to be and why we do this kind of study. And then I'll introduce some like details about the methods and materials, like the data at everything, and then we will go through the results of like what we found in the study. And finally, we can open kind of like discussions about how can we deal with and how can we look into those results and the study as well. So at the background first, like a very big background, I like to introduce a little bit about our lab, and especially maybe Catherine, you could take from there, like introduce maybe something about our lab.
Sure. So our lab in Calgary, we really study brain development. Okay, and with an interest in both typical brain development and atypical brain development. And when it comes to atypical brain development, we look at anything from neurodevelopmental disorders, environmental exposures, brain injury, all sorts of things. But one of my main interests is prenatal alcohol exposure. Long, do you want to go to the next slide where I think you start to get in?
Oh,
yeah. Excuse me. So we've been looking at brain differences in children with FASD for a long time. And we do this primarily using neuroimaging. So we use MRI to get at a lot of different brain features and then to quantitatively assess these. And then we can compare children who were exposed to alcohol to those who weren't. We can look at how these features relate to cognition, behavior, sort of anything else we can measure, we can relate to these brain metrics we get. So we measure gray matter, which is the outer surface of the brain, as well as some of the structures deep in the brain. And then we also look at white matter connectivity, which essentially forms the brain's wiring. And so by doing this, we can get a really comprehensive picture of how the brain is structured and set up and how it's connected and how all of that is working together or perhaps not.
Yeah. So yeah, basically.
Yeah. So yeah, sorry, Long, did you want to take over?
Oh, no, you can talk about this.
Okay, yeah. So we use MRI. So we're scanning kids. And it's always a bit of an interesting challenge. So you see a photo there of one of our kids going into the scanner, and you can sort of see in the background that our scanner actually looks like a rocket ship. We're trying to make the scanner a little bit more friendly and let us get data on a wider variety of kids. The primary metrics that we use to measure brain function come from functional MRI, and you've probably all heard of fMRI, where you see different parts of the brain lighting up. And that's basically what we're doing. We're getting a measure of blood flow and oxygenation within the brain at multiple points. And we can look at how that varies over time and how different parts of the brain are sort of functioning together. And then we look at structural brain connectivity using diffusion tensor imaging, which gives us a number of different measures of white matter connectivity to really get at how well connected the brain is. So you'll hear us talk about a number of metrics from both of these types of imaging today. So diffusion imaging, Long, do you want to explain this?
Yeah, I can. Yeah, sure.
Okay,
Even
yeah,
though
you
you can explain it a bit better. But yeah, here, yeah, since we're talking about like diffusion tensor imaging and white matter, like I showed you before, so white matter, they are kind of like fiber tracks, like they are kind of like wires in the human brain. So we need to find a way to how to capture those like wires or like going through or something, this kind of thing in the brain. We use this technique named the diffusion tensor imaging. Diffusion here means just diffusion, like water diffuse. And you can see that with the different tensor imaging, we can try to kind of like we can capture the molecule or anything like go through this axons, go through those white matter fibers, and generally we use some kind of like a measurement that if the axons or water molecule like go through those like fiber tracks, and then we can say they could have like a faster diffusion, or we can have like evaluate like the white matter integrity and properties, and also if the one molecule like goes through somewhere else, and though they could have like a slower diffusion, and then the white matter measures might be like lower or a little bit like change a little bit. So we use this kind of like a technique to capture the white matter fiber tracks, like those kind of like lines, wires in the human brain, and also and also those white matter fiber tracks or wires are connecting like neurons or cortical to each other. So this kind of like white matter properties kind of like a very fundamental structure in the brain, and also is very crucial, important in the brain and brain development. And also it's kind of like I cannot say vulnerable, but also could be like affected by a lot of like factors during development, something like this. So since today we're talking about the prenatal exposure, and also we just show you some techniques about the diffusion transient imaging, and also Catherine, feel free to chip in if you add anything. So here I'd like to give you some like examples about the previous studies which focus on the prenatal exposure and white matter features, all those like fiber tracks. So on the left hand, those like in this picture, all those like colorful fibers or wires are brain white matters. And you can see all those regions, all those like white matter features or white matter fibers actually present like abnormalities in the children with children with prenatal exposure. And so you can see this kind of thing is kind of happening all over the brain, and also there's kind of like evidence that the children with prenatal exposure present abnormal white matter features measured by the diffusion transient imaging. And on the right hand picture, you can see there's a little bit more like a clear and direct comparison between the children with fetal alcohol spectrum disorder and controls. Or you can see with the part of the white matter fiber tracks, the children with FASD looks kind of like not intact. It looks kind of like a short of some kind of like white matter fibers in the like a back part, like a posterior part of the brain. But however, in control, you can see this kind of like a nice, fully developed white matter fiber tracks in the brain. So you can see that in the previous studies, a lot of studies focus on the white matter changes in children associated with prenatal exposure, or in children or adolescents with prenatal alcohol exposure. So just want to tell you there have been a lot of studies like using this technique and also on this population. And then since we're talking about the brain, like think about brain, of course, like our behavior, like the moment I'm talking, the moment I'm moving my hands, all controlled by brain. And this kind of like a change in brain could have some like adverse or outcomes to the behaviors and cognitive functional behaviors. And one of the way of measuring like behaviors, or sorry, maybe I should rephrase that. Like one of the concept of the behavior is named internalizing and externalizing behaviors. And generally, the internalizing behavior means like some kind of like a problematic behavior like inward, including some like subdomains such as anxious, depressed, withdrawal, or somatic complaints. And then compared to the internalizing behavior, there's some externalizing behaviors. And generally, these kind of behaviors are outward. And they include some subdomains such as rule-breaking behavior and aggressive behaviors. So yeah, in some kind of like a psychopathological or some behavior measurements, we have this like a concept of measurements could measure such problematic behaviors. And also there are a lot of studies also focused on those like internalizing behaviors and externalizing behaviors associated with PAE or in children or adolescents with prenatal alcohol exposure. So again, like I'm going to show you some like previous studies, and one of you can see that there's some like a study also when they look into the externalizing and internalizing behaviors, like in the study on the left from the same this is study also from our lab from the previous PhD student, and they found that the children with prenatal alcohol exposure present increased like higher externalizing scores than control. Oh, by the way, so for those measurements of scores, generally the higher of the scores, higher of the internalizing or externalizing scores means like worse problematic behaviors. So higher is kind of like more problematic behaviors. So and luckily in this study, they found that the children with PAE present higher or increased externalizing like outward behavior scores than controls. And also similar findings in the same data set we're going to use in this study. We're going to introduce this data set later. And also in this study in 2020, they also found that the children with prenatal exposure present increased like internalizing or externalizing scores, and also in several like subdomains. in children with prenatal alcohol exposure. So those two studies already look into this kind of things, and they found, okay, the children with prenatal alcohol exposure present increased internalizing or externalizing behaviors. So so now maybe we already introduced you both the brain, like white matter, and the behaviors. So of course we're gonna investigate the relationships, the correlational relationships between brain and the behaviors, because the behaviors come from brain, so something like this. And again, like in this study, like in the same the first author also from our lab, she investigated such a relationship between the brain white matters and externalizing scores. But in unexposed children, in typical developing children. And so in this picture, you can see this is one of the white matter fiber tracks in the brain, something like in the brain. And they found that in unexposed children, like in typical developing children, they found this kind of like decreased, like from like up to down, like a decrease in negative correlations between the externalizing scores and this one of the measures of the white matter fiber tracks integrity, and it's called the functional anisotropy. I'm going to introduce later, but here the functional anisotropy is kind of like a general measure, one of the measure of the white matter integrity, and generally, but not precisely, but generally, like higher of the functional anisotropy or better means they have like a better development, and so higher of the functional anisotropy is better. So this kind of like an anti-correlation or like negative correlation means that higher of the functional anisotropy, the lower of the externalizing scores. So in this case, it means better white matter integrity means lower or less problematic behaviors. So it's kind of like makes sense, right? So if we have like a better white matter structure, so we have like less problematic behaviors. So they have like, so in this study, they found this kind of like really interesting, meaningful relationship between brain and behaviors. So so before we jump to our next, like for all those kind of like previous study I introduced, and however, there's so far, at least before our study, I don't recall any studies to try to investigate the relationship between behavior and brain. And also one thing I'd like to mention is that among all those kind of like prenatal alcohol exposure studies, like I introduced before, and generally, because those studies are really focused on the prenatal alcohol exposure or fetal alcohol syndrome. So they enroll participants from the clinical places, something like that, and then those children are confirmed or diagnostic already with fetal alcohol spectrum or prenatal alcohol exposure, and also since they are enrolled from the clinical places, so they have kind of like not a very low amount of prenatal exposure compared like something like that. Like they have like more than 1 drink per day or even per week during pregnancy. So I just screenshot one of the tables from one of the study in 2015. You can see in this table, for HC here means controls, PAU means children with prenatal exposure, and you can see the mean alcohol ounce per alcohol drinking is more than like 1 or 2 or even 4 like per week. So it's kind of like a so it can give you some like a hint about like what kind of like what is the level of the drinking quality in the previous prenatal alcohol exposure studies. So so that's kind of like the either limitations or either the kind of like the limitation of the previous study, that if we want to really look into the prenatal alcohol exposure, generally we can kind of like only enroll the participants with a certain amount of prenatal alcohol exposure. But however, we don't have really great chance to really get like a low amount of prenatal exposure, and because also the prenatal alcohol exposure status is very hard to record it, so it's really tricky to do that. And also besides prenatal alcohol exposure, like maybe most of you already know that. Maybe Catherine, you can talk about this, like since this is your study.
Yeah. Sure. Yeah, I can jump in. So I think Long did a great a great job of highlighting how the previous studies that are out there, certainly with regard to brain and also more generally, focus on kids with heavy exposure. And so they've provided a huge amount of useful data highlighting what brain alterations are associated with heavy prenatal exposure. But, you know, for me personally, having gone through pregnancy, for me having lots of friends who've gone through pregnancy, you hear all sorts of things about how small amounts are safe. And as Anna sort of mentioned in the introduction, I think there's a lot of conflicting information out there, particularly for the general public, regarding low amounts of exposure. But it's a difficult thing to study. It's a difficult piece of information to get, and then it's hard to recruit people specifically with low exposure. And then the second issue that a lot of the prior studies have, certainly the brain studies, and I think again more generally, is that there are other exposures that these kids have in their lives. Certainly from our studies that we do in the lab, most of the kids we're looking at are not living with their biological families anymore. So they've had some toxic stress sort of after birth often. They may have other prenatal exposures, so other drugs or tobacco, for example, cannabis. So we know that these things all contribute to brain and they all contribute to behavior, but it's been really challenging in most of the studies to date to actually separate those effects of other types of exposures, prenatally or postnatally, from the prenatal alcohol exposure. So these are the sort of two key questions that we really went into this study asking, and I think you have that on the next slide, Long, is that right? Is that, do you want to go to the next one? Yeah, perfect. Is sort of, what does low level prenatal alcohol exposure, how does that affect brain and behavior in children? And how can we separate that, or can we separate that from some of the other factors that we know affect brain and behavior in children? So we kind of came into this study wanting to investigate both white matter differences, so that's brain connectivity differences, behavior differences, and then also the relationships between the two of them. So how do the brain abnormalities potentially contribute to the behavior problems that we also observe?
Yeah. Yeah, and also the chat, like for myself, like maybe also kind of, I'm happy to share the slides, and maybe the CanFASD could find a way to share the slides, I guess.
Yeah, that's fine.
Yeah. Right. So those are kind of like two questions in our study. Yeah.
Mm-hmm. So we took advantage, Long, I don't know what's on your, do you want to go to the next one here?
Oh, yeah, sure.
Yeah, so we took advantage of this great resource in the US, which is called the ABCD study, or the Adolescent Brain and Cognitive Development Study. And it's a huge study that's collecting brain and behavior data at multiple sites all across the US. They have over 10,000 participants, all recruited at about 9 years old. And they have imaging data, and then they have really comprehensive prenatal and postnatal health histories. It was acquired retrospectively, which is a limitation, but they have this really rich data set. And so nobody in the study was recruited deliberately for prenatal alcohol exposure, but just because there are so many participants, we were able to take real advantage of this and select the participants whose biological mothers reported alcohol consumption during pregnancy. And Long, I'll let you explain a little bit more about the specific methods.
Oh, yeah, sure. So yeah, just like Catherine mentioned, this ABCD study is it's a very big study, and also it's from the United States, not from Canada. But in future, of course, we're gonna have, maybe we can also have this kind of thing happen in Canada, and also like I mentioned before, so this study involved in both brain neuroimaging and cognitive function and behaviors. And the participants enrolled here are the adolescents, so they are like kind of like 9 to 10 years old. And also it's a long-term study. So in the present study, in the study we have like today, we're going to show you is only focused on their baseline study, like the first time they scan. But also in future, there are going to be like more data for those participants. So yeah, like Catherine introduced, we have like diffusion tensor imaging. I said before, or something like in the chat. Yeah, okay. So And so we can use diffusion tensor imaging to investigate white matter fiber tracks, and also they have like questionnaires. We can check about the prenatal exposure status, and also some like a social economic status, and also the internalizing and externalizing behavioral measures. So next slide. So here you can see, here I can go through a little bit about the prenatal exposure status in this study. So here are the two screenshots I took from their data sharing website. So you can tell on the left hand, on the left hand of the picture, you can see the question is kind of like in structure of beginning with before knowing pregnancy. So they ask the parents, like before they knowing pregnancy, whether they took several adverse substances, including tobacco, alcohol, and also other substances such as heroin, cocaine, cannabis, etc. And also then on the right-hand side, of the questionnaires, again they ask the parents when they are knowing of pregnancy, whether they took tobacco, alcohol, etc. And also in alcohol, you can see they have kind of like nicely, they have also some questions about the quantity of alcohol, such as like maximum drinks in one sitting or maximum drinks in like also average drinks per week, something like this. And also, like Catherine mentioned, in order to get our data kind of like reliable, we only took those questionnaires from the biological mothers who answer those questions. So this is the first thing we want to make sure the answers from those questions are reliable to investigate prenatal alcohol exposure or prenatal exposure status. And also, in order to get for more reliable of our participants with prenatal alcohol exposure, we only took okay, cool. We now have a circle. So we only took the question from this one, from the knowing of pregnancy alcohol, means that if the biological mother answered yes in this knowing or pregnancy, whether they took alcohol, answer yes in this question, and then we consider these participants with prenatal alcohol exposure. And also, of course, again, like also we excluded mothers who also answered yes in other substance exposure, such as tobacco, heroin, cocaine, cannabis, etc. So we want to make sure the participants and adolescents be selected in the study that they only exposed to alcohol, and also only exposed to alcohol when mothers be aware of pregnancy. So we want to make sure the participant we have, all the data all the analysis we have in later gonna be reliable and meaningful. And of course, like Catherine mentioned, said, those questions are took from the biological mothers, and their children, their adolescents, are already 9 or 10 years old. So they kind of like they need to get their memory back to remember what happened during pregnancy. So even though there might be like a limitation, so these questions might be not 100% accurate, but this is the best we can get at the moment. And also, in this kind of study, it's always tricky or hard to get the really precise prenatal exposure status. Yeah. So okay, so in this table, you can see like Catherine introduced before, this study have over 10,000 adolescents and participants. But however, after we remove all those kind of like criteria, make sure the questionnaire answered by biological mothers, and also make sure the children or adolescents with prenatal exposure only exposure to alcohol but not to other substances, then finally we have 135 participants with only prenatal exposure to alcohol. And also, yeah, this kind of, then we have this kind of like, but still 135 is still kind of like a meaningful sample size. And you can see the age is around 10 years old, so they're all adolescents, and likely the sex balance is kind of a balance between female and male. And the total family, and also total family income, we'll talk about later. But first, let's look at, like I mentioned before, in this questionnaire, they also ask the drinking quantity during pregnancy. So when we're looking at the drinking quantity of pregnancy, you can see the drinking quantity of pregnancy of those participants with PAE is quite low as compared to the previous study I showed you before. And this amount is around like 1. Somebody may even like write down like 0.5, something like that in this question. So they are quite low amount of prenatal.
Just to reiterate here, I think this is really important because we have a very clean sample as far as things go. So low exposure, just 1 drink per week was average, and they were matched with the controls on family income, on parent education. Everyone was still living with their biological parents, so nobody had switched caregivers, same age, same sex distribution, and nobody in either the control or the PA group had other exposures. So really as clean as we could possibly get here.
Yeah, that's how we select the controls. Like we make sure like other factors such as age, sex, like a family income or parent education is very balanced or very matched to the children with low amount of prenatal alcohol exposure. And also, by the way, so the value here, the 9 here means the income is between the $100,000 to $200,000. So they are kind of like from the middle class kind of like families. And also for the parent education here, the value 18 represents the parent, the biological mother has the bachelor degree. So also as they have a kind of like a certain level of education levels. And also we select the participants with a similar income and education, because like I mentioned before, in this study they have over 10,000 participants. We don't want to select like 9,000 participants as the other group, and then we compare 100 children with PAE to like 10,000 or 9,000 controls. We want to make sure, this kind of like 2 groups were selected like balanced and also statistical meaningful, and also without any other factors affected. But of course, there are more other factors we cannot imagine. Maybe we didn't imagine, and also include the data set. But in this study, we just try to balance like age, sex, like also the social economic status to make sure those 2 groups are balanced. So we also the reason why we want to do that is that the results I show you later, they should purely from the effects of prenatal alcohol exposure, not come from the other things, hopefully. So the data, so then the data we took from this study is just like I mentioned before. So first we took the diffusion tensor imaging data, which is a measurement of white matter, and we used some measurements of this diffusion tensor imaging named functional anisotropy, which kind of like the measure of the white matter integrity. So higher of the FA, generally higher of the FA, it's better. So just you can just assume that, and then we use this child behavior checklist scores to get the internalizing and externalizing behaviors. And in this scores, however, the higher scores means more problematic behaviors. So it's kind of opposite to FA. So. And then additionally, like we discussed, like say something like introduction. Now we have brain and then we have behavior. Of course, we're going to investigate the relationship between brain and behavior. And we hypothesize that the relationship between brain and behaviors might be also affected by this low amount of prenatal alcohol exposure. All right, so that's all the data I introduced, like we have from the study, and all the data I introduced before. So first I'm going to show you, the first thing we did is we just simply compare those behavior measurements between the children or adolescents with low amount of prenatal exposure and matched unexposed controls. And you can see in the column here, those are the domains of externalizing and internalizing behavior scores, and the first 2 are the summary scores of those domains, and those scores are kind of like sub-scores of those of related domains. And here are the values of scores of PAE, like children with low amount of PAE, and here are the scores of matched controls. And you can see, and if you're familiar with statistics, you can just jump into the p-value here. And if you're familiar with statistics, you can see, those scores of those 2 groups actually kind of like similar to each other. So it seems like the low amount of PAE kind of like did low amount of PAE didn't affect externalizing or internalizing behavior that much, like not seriously changed like we showed in previous studies. But however, we still spot this one summarized score, like externalizing score, that even the children with low amount of PAE showed higher scores than matched controls, and it can reach to a certain statistical level. So it means that even the children with low amount of PAE, they might still have some kind of a potential problematic behavior changes in this case, in this like population. So it doesn't mean like, okay, even though there's low amount of PAE, the behavior are still all the same to controls. But however, at least in this data set, we still found this one significant change in the externalizing summarized scores. So low amount of PAE. So in here, like to say, like low amount of PAE still have, at least we found that low amount of PAE still affect behaviors in a way. Okay, so this is a pure behavior, so no brain. And then let's jump to the brain. And also luckily in the measurements, we luckily also found that the decreased functional anisotropy in several regions all over the brain. So this is the left part of the brain, left hemisphere, and this is the right hemisphere, like a brain. And those nodes, like those blue points, are the location of the brain, not the shape of the brain. Of course, there's no way our brain looks like this, but those are the location of the brain. So you can see the location of the brain is somewhere. Those regions are somewhere around like the post part of the brain, around the motor cortex, like and also the attention cortex, around the temporal cortex, and also some regions around the occipital, like the back of the brain, like which relate to the visual functions. So it means that it means that because in the hypothesis, maybe all in our like general findings, that the children with low amount of PAE wouldn't have like brain changes because of the low amount. But however, in this study, we found a decreased functional anisotropy, which means like affected white matter integrities still like in children with low amount of PAE. And also this results, those results are kind of like similar in term with the things I showed you before. Like if you remember all the fiber tracks, and in the previous study, they found that the children with PAE showed a decreased white matter fiber tracks, decreased functional anisotropy, or in affected white matter fiber tracks compared to controls. So it means that even in the children with low amount of prenatal organ exposure, we still find this the similar findings which we found in the like typical a certain amount of children, a certain amount of PAE in previous studies. So yeah, so at the very beginning, so it's kind of like interesting to see, because it should be those regions shouldn't show any difference, but still they showed up and showed a decreased functional anisotropy. Okay, so since I introduced you the behavior and the brain, and then of course, before we jump to relationship, this is kind of like this picture is kind of like the give you like more details about this change in those brain regions. So it means that not only look to like one single measure of the white matter fiber tracks, but also when we look at the different kind of like measures of white matter fiber tracks, and even a little bit, even one of the measures of the functional signals, we can see that rather than the functional anisotropy I showed you before, some other measures also showed change in children with low amount of PAE. So here, the blue color here, you can see these are the kind of the measure of the control, and the red color here are the measurements of the children with low amount of PAE. And ideally, ideally or hypothetically, those two lines or those two things should like overlap to each other. But however, you can see they're not overlapped, and there's some kind of like a gap between blue lines and red lines. It means that there are some like changes in those white matter measurements between children with low amount of PAE and exposed controls. So those kind of like changes are kind of like solid in the present study. Yeah, and also those, if you're familiar with statistical stuff, like those the findings, the regions, all those functional changes, the changes in those functional anisotropy could also fall into some very stringent statistical power. Yeah. All right, so let's jump into the relationship between brain and behavior. So like I mentioned before, like if you still remember, like maybe I should jump to the slide, but I still remember in the previous studies in the typical development children, they found negative, like from up and down, like a going down relationship between externalizing or internalizing behaviors and white matter measurements, which is we kind of like replicated these results in the same study as well. So the blue line and the blue dots here represent controls, and the red line, yeah, maybe I'm sorry for anybody who's not like colorblind, but however, you can just remember that this kind of line with a little bit angle here are controls, and this kind of line with a little bit horizontal, it's the data of PAE. So you can see that in typical development children, we have this kind of like decreased relationship that, okay, the higher of the functional anisotropy or higher of the white matter integrity, lower of the externalizing scores. So it kind of like makes sense. Better white matter structure, lower of the or less of the problematic behaviors. But however, we didn't find such relationship in children with low amount of PAE. You can see the line here, the horizontal line here, or the red line here are kind of horizontal. There's no such a relationship there. So it means that even with low amount of prenatal aqueous exposure, this kind of like a brain and a behavior relationships, like interactions, still affected. So it means that like there must be some kind of like altered way between like a brain and behavior, like something happened in the brain and then something happened in our behaviors. Such a relationship is also like change to each other. So it also is kind of like a very interesting findings in this low amount of prenatal aqueous exposure children. Yeah. Okay, so yeah, that's kind of like all the results we have. So first thing, so if you remember, first we found the increased externalizing scores or more problematic behaviors in children with a low amount of PAE. And then also we found similar white matter changes, white matter fiber changes in the previous PAE studies that the children with low level of prenatal exposure present a decreased white matter integrity. And then again, furthermore, we found these altered relationships between brain and white matter or brain behavior and behaviors. So it's kind of like also kind of like interesting to see this kind of results, and also here's some kind of like discussion. Yeah, maybe Catherine, you can just
Sure. Yeah. So I mean, I think this work is really interesting because, to our knowledge, it's some of the first evidence that low levels of PAE affect the brain and also disrupt this brain behavior relationship. So I think it's really important to get this information out there because we finally have something we can point to. Instead of the recommendations that say there's no safe amount, we can actually provide evidence that low levels potentially have an effect on brain and behavior as well. So I think it provides more robust evidence than we've had before. Certainly for me, it answers a key question that I had through my many years of PAE research to date, which was about what low levels do. So I think there's a lot of value in this kind of a finding, but there's also a lot that we still need to do. And so one of those things is to look longitudinally. So look how these kids' brains are growing with age. And we know from studies of heavy PAE that there's not just brain changes, but there are differences in the way the brains grow in kids who've had heavy prenatal alcohol exposure. So we want to look at these kids with low levels of exposure over time and see whether their brains are growing differently than the brains of kids who are unexposed. And then of course we want to, learn more about the timing of exposure and the amounts of exposure. And there's always more questions at the end of a study like this than there are answers. But I still think that this was a really exciting study to me. I was really excited to be able to do this and to be able to provide some evidence. So that's our work. And I see one question at least popping up in the chat. And I think, Long, I don't think we have any more slides, do we? So I think we're happy to take any questions that anybody has.
The final slide,
okay, yeah.
It's final, but still very important slide, actually. This is very important. Catherine, you can.
Yeah, so thanks everyone for listening to us today. So yeah, I'd like to thank you so much, Dr. Long, for staying with us for the Q&A. and for this awesome presentation. And I'd like also to thank all of you who joined us today, and hope you will join us in future webinars. So just a reminder that both SPI and CanFASD host a series of webinars of interest to clinicians, caregivers, and service providers. So if you want to get more information of upcoming webinars and to view past webinars as well, you can go to skprevention.ca and click on the Learning Opportunities tab. And also you can find information on CanFASD webinar series at canfasd.ca and at the CanFASD YouTube channel. You can follow CanFASD on Twitter, you can follow the CanFASD blog, and other their social media channels to be aware of the latest research, resources, and events. So many thanks to all of you, and I hope to see you again soon. Bye-bye.
Thank you for your time and listening. Bye.
Thank you so much, Dr. Long.